So thank you all so much for joining us today. And I am so absolutely thrilled that we have Dr. Tariot with us from the Banner Alzheimer's Institute. My name is Emily Kile, and so we try to host these about once a month. So I very much appreciate you all being here. It is being recorded. And if you have questions, if you can type them into the chat box or the Q&A at the bottom of my screen anyway, and I'll keep an eye on those while Dr. Tariot does his presentation. So we can be as interactive as possible. We will go for an hour, so I apologize if not all of your questions get answered, although if we're able to get back to you, we certainly will. And so with that, I'm going to let Dr. Tariot introduce himself, and then we'll get going and we'll be done promptly at 1 o'clock. So again, thank you all so much for joining us today.
Okay, thank you, Emily. And Emily's resume that she probably doesn't think about includes bringing my wife, Laura, and me to the Valley. About 18 years ago, I was referred to Emily to get some help with my contract negotiations with Banner. So we're very grateful and have very fond memories of our interactions now, almost 2 decades ago. So I'm an internist, geriatric psychiatrist, clinical trialist. I do some translational research. I'm part of a team of about 200 people at the nonprofit Banner Alzheimer's Institute, where we provide outpatient care on a consultative basis to persons and families either struggling with or faced with the future of diseases like Alzheimer's disease. And then we do a boatload of research. I'll give you a glimpse of some of that. This is going to be a slightly researchy oriented talk, but hopefully plain enough English. I still see patients and I still do hands-on research, and I love what I do. So a common question, even from doctors, even after I've given my talk, is what's the difference between Alzheimer's disease and dementia? So dementia is a general term. It's a syndromal term. It's like saying pain or cancer. If you told me I had cancer, I'd say, which one? What do I do about it? What about the genetics? Dementia refers to progressive loss of memory and other thinking ability. It has to include erosion of the ability to function normally on a day-to-day basis. Almost always includes changes in emotions and temperament. And frequently there are neurological manifestations such as balance and gait problems, continence difficulties, sleep-wake cycle disturbance, seizures, and so forth. That's dementia. It's actually not really a diagnosis. It's a descriptive term. There are maybe 100 or so different illnesses that can cause that picture. But as it happens, Alzheimer's disease, and we'll get back to this a little bit later, is a specific disease that causes at least 60% or so of all cases of dementia. So people can get them confused. So when we talk about Alzheimer's dementia, this is the clinical characteristics. Creeps in on little cat's feet, very subtle changes, usually beginning with memory, but not always. Sometimes language, sometimes visuospatial function. There has to be change in at least 2 areas of cognitive function. I give some examples here. Again, I'll emphasize this functional impairment. And you have to be sure that there isn't some other medical culprit causing this picture of failing brain health. I teach my trainees a mnemonic, vitamin E, to be sure that we're thinking properly about the other potential medical causes. Is it vascular, is it infectious, traumatic, autoimmune, and so forth. So you do have to do that, but this is a classical kind of picture of what Alzheimer's looks like. When I'm teaching trainees, I give them my idiot's guide to the differential diagnosis. Work with me here. Down at the bottom, you see roughly 2/3 of all cases of dementia will be due solely or primarily to the disease, Alzheimer's disease. We'll get back to the science of that in a minute. If you move to the left, you start getting into dementia due to blood vessel problems in the brain, sometimes co-occurring with Alzheimer's disease pathology, sometimes purely vascular. Not as common as many people think. This may be too into the weeds for you, but I can't help myself. A lot of primary care doctors think that vascular dementia is far and away the most common culprit. That's really because they do a nice evaluation. They might get a brain MRI scan, and the piece of paper will say, there's some changes here consistent with microvascular disease. We call these white matter changes. They're not. They're caused by something else. So that's a common misunderstanding. Move to the right, we get into a really gnarly group of brain diseases that are basically Parkinsonian disorders. Parkinson disease itself, or a rather sinister cousin called Lewy body dementia or Lewy body disease. These are essentially, they can cause dementia. They have Parkinsonian features, but they're different from Alzheimer's disease. And then over to the right, and I'm not going to get into details here, is a group of disorder that we call the frontotemporal dementias. Rare, malignant, not treatable other than just helping people manage the condition. The two key forms or presentations are the language variant, somebody whose memory may be pretty good but can't either understand the spoken word or can't speak, or the more troublesome variant we call the behavioral variant, where somebody's cognition may be pretty good but their behavior is floridly bizarre. I don't know if Rod Blagojevich has frontotemporal dementia, but I think of a true case. I just want to give you a vivid case example of an OBGYN in New York City who successfully completed a very complex operation on his patient and then took out his scalpel and carved his initial in her belly. That's the type of florid, bizarre behavior that this condition can present with. So, to put it a little more simply, Alzheimer's far and away the most common culprit. We have to think about vascular burden. We have to think about the folks with Parkinsonian presentations, and then the rarer frontotemporal presentations. So now you are a diagnostic expert. To complicate things a little bit, as we age, the odds go up that if we have a dementia, we're more likely to have multiple culprits, or sometimes even at autopsy, we can't find what caused it. So, you know, by the time somebody's in her or his 90s, very like 50-50 chance anyway of having more than one cause of the clinical presentation. I'm going to back up a second. There's a tricky little detail here, which is clinicians are making probabilistic diagnoses of pathological conditions. You wouldn't do that for cancer, but in this business, we don't do brain biopsies casually to establish the actual pathology. So as I'll get to, we now have better and better tools to do this without cutting your skull open. So let's say I've got two people, one person that I'm following over time and one person who's waiting in the lobby to see me. In both cases, I'm going to be assessing that individual's Venn diagram of how they're doing cognitively, functionally, behaviorally, and neurologically. And one of the reasons experts, I guess I accidentally labeled myself an expert, but anyway, people who do this professionally like it is because every individual's Venn diagram is different. Everybody's different. And the way it changes over time is unique. So you're always dealing with unique and interesting situations. So here's a schematic of, let's just say, my thinking ability over time, knowing that I'm destined to develop Alzheimer's dementia. I will go through many years of normal thinking and memory. But what we now know is that if I'm destined to develop Alzheimer's disease and dementia, there will be a phase where the disease process is ticking away silently, the way it happens with prostate cancer or liver failure or kidney failure. Change is happening, but you don't know it because there aren't manifestations. And those changes that we'll get into in a little bit of gory detail in a minute start occurring as long as 20 plus or minus years before any symptoms become evident. 20 years. I'll get back to the significance of that later, because that's the zone where we think we can make the biggest difference with interventions before the brain is ravaged, therapeutic interventions. Then I'll ease into a phase of this course called mild cognitive impairment. That's a condition that roughly 10 million Americans have. That refers to noticeable decline in cognitive function, noticeable to me perhaps, more likely to my wife Laura, maybe my doctor. So that's one condition, concern. Another condition or a criterion is if I do cognitive testing, I have to perform a certain percentage below my age and education adjusted peers. And this is critical, it can't cause significant erosion in my capacity to, on a daily basis, my functional capacity. Sort of something for you to be aware of is that judgment is actually tricky. So don't judge me about, I don't know how many of you live in Phoenix, but don't judge me about whether I lose my car in Terminal 3 at the airport. But if I'm being interviewed by my doctor about functional loss, he or she may say, well, are you able to keep up with your appointments? And I would say yes. And Laura might say, yeah, but a year ago it was all in his head. Now he's got sticky notes all over the house. He still panics. You know, it's a different scene. Is that functional impairment or not? These are judgments that we need to make as clinicians. Eventually, I will slip into the full-fledged stage of dementia. So we think about this continuum, and it's a seamless continuum. So it's a little hard to put seams and say, today is the day I hit mild cognitive impairment criteria. More likely, that will become evident over a period of a year or two. Let's do a little science here. And I'm going to focus on Alzheimer's as the paradigm of the dementias, obviously not the only one. In the image to your left, you see two brains at autopsy, one from somebody who died at about age 75 without Alzheimer's, and then somebody who died at the same age with Alzheimer's disease. You can see the obvious loss of brain tissue. So that it's a disease that kills brain cells, prunes connections between them, causes deposition of misprocessed proteins. And the big boys you'll hear about are the amyloid proteins. These are misprocessed proteins that deposit outside of cells. I don't know if my pointer is going to be evident to you, but these are proteins that start aggregating outside of cells. And then there's a different type of protein, this ropey, thick, deposited, abnormally processed tau protein. And those ropey deposits are called tangles, and they are inside cells. These are the sine qua non microscopic criteria for Alzheimer's disease. There are multiple other changes as well that I will touch on a little bit later. The picture is actually August Dieter, Alzheimer's patient. These are his photomicrographs. Believe it or not, she was in her 50s when that picture was taken.
Can I ask you a question, Dr. Tariot?
Sure. Call me Pierre.
when you talked about the sort of 20 years may have passed when you're kind of in that pre-stage, and maybe you're going to talk about this later, but is there testing or scans that can be done so that you know scientifically that people are in that pre-stage? Is that how you know that there's sort of this 20-year time frame to see that information?
Yes. And we will get to it.
Okay, sorry, thanks.
No, no problem. I'm happy to keep this casual. A little more science here, and then I'm going to show you a slide from Heck, and then we'll get normal again. The way we think about risk factors for developing Alzheimer's disease are the Alzheimer's presentations that occur quite early in life, and then those that occur later in life. The textbooks say that early onset means onset before the age of 65. Frankly, we think it really should mean before the age of 60. In some cases, it's in the 30s and 40s. The textbooks say that the early onset folks are less than 5% of all cases. It's probably well below that. The headline here is that there are 3 genetic mutations. That is to say, misspellings of the genetic code, a single amino acid substitution at 3 different types that inevitably lead to early onset Alzheimer's disease in the person who carries the mutation and half of his or her offspring, and so on through the generations. We call this autosomal dominant, fully penetrant condition. So, and you may also be aware that persons with Down syndrome by the age of about 35 have a head full of Alzheimer's changes, although they may not be frankly symptomatic at that age. It's quite important scientifically that all of the genetic mutations and then the replication of the gene in Down syndrome all cause amyloid dysregulation. So right there, you get the beginning of about a 30-year scientific story focused very heavily on amyloid dysregulation. Why does it happen? Why does it cause at least this form of Alzheimer's disease? Then we get to the late onset, much more common late onset Alzheimer's folks. This is mostly folks in their 70s and 80s. This is a population-based study. Ignore the grades of gray, white, black here. Just look at the height of the column. If you take all US residents between the ages of 65 and 74, maybe 5% of us will meet Alzheimer's dementia criteria. 75 to 84 hits almost 20%, okay? And then 85 and older, 40 plus or minus percent. So if all I know about my new patient in the lobby is that she's 86, already I'm on my toes that she has almost a 50-50 chance of having dementia. And Emily, I don't know if you want to monitor the chat. Are you able to do that, or am I doing that?
No, I'm monitoring the chat, so I didn't want to interrupt you, but the question came up as you see that since there's a genetic connection, if both of your parents have been diagnosed with dementia, then what's the likelihood that their offspring are also going to have that same genetic issue?
That's a great question. Let me begin by answering it this way. And it depends on which dementia it is or was. So let's say we know that my mother had Alzheimer's dementia, and I'm talking to Emily, and neither of her parents did, and none of her siblings did. My risk is about 300% greater than hers of developing Alzheimer's dementia in the future, although that risk doesn't express itself statistically until the 8th decade of life, until I'm in my 70s. If both parents definitely had Alzheimer's dementia, then the risk goes up even further. But let me get into a little bit of late onset genetics here. The big culprit for late onset Alzheimer's disease, far and away the more common form, is whether or not you carry a version, not a mutation. So it's an important distinction. We talked about mutations in early onset Alzheimer's disease. There's a different type of genetic variation. We call it allele. It's just a physiologic variant that doesn't seem to cause disease. But in the case of this gene called apolipoprotein E, APOE, it comes in 3 versions, 2, 3, and 4. I don't know what happened to 1. And it turns out that if you carry 1 or 2 copies of the 4 version, your risk of getting Alzheimer's disease late in life goes up, but it's not certain. So it's not causative the way the mutations are causative. So if I carry one copy of the APOE4 variant, which a quarter of the world, for the most part, does, my risk of getting Alzheimer's disease later in life jumps up about 300%. If I carry two copies of the APOE4 gene, my risk goes up about 35-fold. It's not certain, but it does really jump way up. And then it's important to note that the converse is true, that if I'm lucky enough to carry a version called 2, APOE2, my risk is significantly reduced compared to somebody who doesn't carry a 4 or does carry a 4. And then another genetic pearl or jewel is that a very rare genetic mutation was found in women in Iceland that blocks amyloid dysregulation and prevents the emergence of symptomatic Alzheimer's disease, even in women who carry the APOE4 variant. So once again, there's this critical genetic evidence that amyloid dysregulation is at least a key player here. And we'll come back to that. I'll come back to lifestyle factors a little bit later. Here's my slide from Heck, but I'm gonna make it simple. I wrote a paper some years ago trying to make sense for me about how is it that there can be all these factors that either increase or decrease risk. How do you make sense of that and end up with this sort of final picture that is classical symptoms, classical plaques and tangles and nerve cell death in the brain. So across the top, there are genetic factors that can increase or decrease risk. There are environmental factors that can increase risk. Don't become a professional boxer, don't cliff dive, don't inhale insecticides. Age, we talked about already, is a big factor. There are lifestyle or endogenous factors that increasingly look to be important for either increasing or decreasing risk, and we'll come back to that. There may be exogenous protective factors that as yet are unproven, estrogen, antihypertensives, anti-inflammatory drugs. So each of us marches through life carrying or experiencing these risk increasing or decreasing factors. But if I'm gonna get the disease, I'm gonna hit a tipping point where a molecular domino effect is unleashed. And I'm not gonna get into the details, but bad things start going wrong at a very fundamental level. Stress response mechanisms kick in, but they fail. And then eventually you start seeing these proteins being misprocessed. That's the sort of earliest so far that we've been able to identify the disease unfolding. And then eventually the full-fledged picture of plaques, tangles, neuronal loss, symptoms, and death. So this is actually an important slide in a way, but just think of it as a visual. If you want to imagine what's going on in the diagnostic and treatment research world, you could just sort of pick any level of this picture and people are devoting their careers to studying different aspects of this. To Emily's point a couple minutes ago, we now have the ability to detect many of these changes, the most critical ones anyway, very sensitively, decades before symptoms emerge. If you start with me at noon, and we'll go around the horn here, around the clock, I'll get to this in a minute. A huge advance in the last year has been the advent of blood-based biomarkers that, believe it or not, are as good as a biopsy, apparently, at establishing the presence of Alzheimer's pathology, as well as the presence of nerve cell degeneration and death and inflammation. So this is huge. We can probably substitute these blood biomarkers soon for most of the other tests that we've been using, including spinal fluid testing, which can give the same information, but it's trickier to get. move over to sort of 3-ish, we get into the MRI variables. We can measure, that's magnetic resonance imaging of the brain. We can measure brain structure, volume, and we can use elegant techniques to look at pathway integrity and actually even molecular functioning. About 5 o'clock, we have our traditional measures of thinking and functioning and behavioral manifestations. 6:30 or 7, we get to the genetic tests. And then over to the left, we can use positron emission tomography to image amyloid protein deposits in the living human brain and tau and tangle deposits very sensitively. But and then I'll just mention that we look at glucose activity in the brain too with PET scan. That's going to sort of wane in its importance. But I just want to underscore these fluid biomarkers. Sorry, this is so wordy, but there was a front page above the fold article in the New York Times when these papers came out last summer, and I forget the headline, but it was something like "Unbelievable" was the headline, something like that. The takeaway is individual blood tests can tell us whether Alzheimer's pathology is present or absent in the brain. So this isn't commercialized yet, but it probably will be in a couple of years. And this is going to blow the lid off diagnosis as well as tracking treatment effects. And you'll be hearing a lot more about this in days to come. I'm more than half done in case you're starting to fall asleep, so stay with me. We're going to get excited about treatment research. In the Q&A, we'll have plenty of time for more Q&A, so if you have questions about current FDA-approved treatments for Alzheimer's disease, we can get into that. There are basically 3 in one class and 1 in another class that are approved for treatment of symptomatic Alzheimer's disease, and they primarily may bump up memory function a bit or other aspects of cognition, but primarily serve as a slower downer of disease progress, symptomatic progression. New treatments have all these goals. I'm not going to read them, but you can imagine that a drug designed to improve my brain functioning in the moment through, for instance, optimizing chemical messenger functioning is going to be a completely different type of treatment and treatment objective than a drug that doesn't do anything to how my synapses are firing now, but is designed to block further damage to my brain cells, a so-called disease modifier. So I will mention now that there are 126 experimental agents for treatment or prevention of Alzheimer's disease in clinical trials as we speak. And they have vastly different purposes and mechanisms, as I'm trying to suggest here. And a big question is, when should you start? And I'll get back to that later. These are, of the 126 agents being tested, these are kind of the themes. Can we go after amyloid? Can we go after tau? Can we fire my afterburners, get my neurons in my brain working better? Can we block rust? Can we block cell damage? Can we give Arnold Schwarzenegger kind of therapy to beef up the cells that are surviving? Can we block inflammation? I won't get into it unless you want to in the Q&A, but there are intriguing hypotheses that certain types of infections may fan the flames of Alzheimer's pathology, not cause the disease, but accelerate it. So some anti-infectives are being looked at. So I'm going to just focus on a couple of key examples before we wrap up. Amyloid is definitely a key target, and I'll just kind of recapitulate the reasons. I've told you that amyloid plaques that you see under the microscope are a sine qua non for establishing the presence of the disease. bits of amyloid are known to be highly toxic. Now that we can do PET scans and blood tests to assess amyloid burden in the brain, we now know that if I'm walking around with a head full of amyloid, I'm at much greater risk of having frank Alzheimer's symptoms in the future, although it doesn't guarantee it. I mentioned that these mutations that cause at least one form of Alzheimer's disease inevitably cause amyloid dysregulation. I mentioned the Icelandic mutation that blocks amyloid dysregulation and therefore blocks the emergence of the disease. So for all of these reasons, there's been tremendous interest in going after amyloid.
So, Dr. Tariot, there was just a question while you're there.
Oh, yeah.
So the person says, recently COVID researchers have partnered with Alzheimer's research due to brain scans post-COVID looking similar. It's been suggested post-COVID can predispose one to developing some form of cognitive impairment. So have you seen any of that research on what those brain scans look like and whether that looks like Alzheimer's or is Alzheimer's, or we just don't know anything?
Yeah, and I'm just going to do a sanity check here. Oh, we're okay for time. So this SARS-CoV-2 virus is really nasty. COVID, of course, is the clinical disease picture that it causes, but that's the virus. It is known for certain to cause brain damage in some people, in some cases immediately, in some cases it's delayed. There's work being done. It's likely that in some people, the virus directly infects the brain, possibly through the olfactory bulb. There's also evidence that it may get into the brain carried by blood instead of direct invasion or infection. It binds with certain receptors in the brain that cause mischief. It causes coagulation problems leading to strokes, full-blown large strokes or micro infarctions. Some of these changes are visible with various brain imaging techniques. I think that quite a lot of grant support is going toward understanding the mechanisms of damage to our bodies of this virus, short-term and long-term. There is, I would say, a moderate degree of concern that particularly for people who have cognitive dysfunction early on, that they may be at greater risk for subsequent brain health problems, accelerated neurodegeneration. It's not to say that I'm suggesting it causes or accelerates Alzheimer's disease, but there is concern and there's a lot that we don't know. So we can, if that wasn't satisfactory, we can come back to that important question later. So here's a little cartoon. The lower left says cell surface. I guess that looks like a piece of carpet or something. That's the cell membrane. The deeper color to the right is inside the cell. The rest is outside the cell. The way amyloid goes wrong is there's this thing called APP, amyloid precursor protein, that dangles both outside the cell membrane and goes inside the cell. We all carry this, it's normal. But normally what happens is that there's a pair of scissors called enzymes, enzymatic scissors that snip that precursor protein in a certain way, and this SAPP beta, that's the non-harmful form created by a certain kind of scissor cut, no harm, no foul. For reasons we don't understand, but at least are partially genetically driven, in Alzheimer's disease, two different pairs of enzymatic scissors clip that precursor protein twice in the wrong way, leading to highly, highly toxic fragments that sort of instantly cause mischief and cell death. And they gradually aggregate into clusters called oligomers, and then more dense structures called fibrils, and then amyloid plaques. So the smart women and men in the labs have figured out how to interrupt every single aspect of that cascade. It's unbelievable. It's not as great a story as the COVID vaccines, but it's a fabulous story. So we have antibodies against essentially all the soluble and even insoluble forms of amyloid. We have oral medications called secretase inhibitors that can block the clipping. And so billions of dollars have gone into studying these agents over the last several years, including now. I'll give you a case example. It may not be obvious at first why it's so compelling. So work with me here. I can't read the title because it's blocked by my header here, but what I want you to look at, so these are amyloid PET scans. And let me block my pop-up. Amyloid PET scans. So column to the left, baseline. Next column is 1 year. As we go down, these are pairs of PET scans in people a year apart who were in a trial of a monoclonal antibody made by Biogen called aducanumab or aducanumab. And what I want you to see, just look at the first pair at the top. This is somebody who was treated with placebo. And you see, if you just scan quickly from left to right, that person's pair of amyloid PET scans looks about the same. The next person on the left, you see a lot of orange. On the right, a year later, much of the orange is gone. The orange depicts the amyloid deposits. And the next pair is somebody who got a slightly higher dose after a year, much of the amyloid was gone, and so forth. So on, so forth for the last pair of scans. So this was the first proof of principle that you can remove these pathological gooey protein deposits from the living human brain in persons with Alzheimer's disease with a monoclonal antibody. Big deal. This was actually a very small study. It led to a global program studying aducanumab that has had a remarkably convoluted course. The bottom line is 2 global studies were underway. An interim analysis was done suggesting that maybe there wouldn't be clinical benefit. It was declared futile. They gradually wrapped up the trial over months. And when all the data came in, they said, whoops, it's working. And so this has posed a real dilemma for the FDA. We can get into the weeds with that if you want. So the drug is now, an advisory committee reviewed the data and fussed a lot. The FDA has announced that they'll make a decision about possible approval for marketing this June. If approved, it would be the first disease modifier for Alzheimer's disease, a real landmark if it's approved. I just want to mention that we're further behind with therapies addressing tau and tangles. I don't think I'll get into the details here, but this is a major development, and you'll be hearing quite a bit about the tau-directed therapies. So far, none of them have panned out. The details matter a lot about exactly how the drug or biologic is targeting the tau and tangle pathway. But this is going to be a big deal because despite all the yammering I've been doing about amyloid burden by itself or even clearance doesn't correspond with clinical manifestations. But there's a very direct correlation between misprocessed tau protein burden in the brain and symptoms. And there's this dance between amyloid and tau that goes on that we can come back to a little later if you want. But so tau-directed therapies are commanding a lot of attention. And if you're still awake, you're already thinking, well, gosh, guys, why don't you combine them? So that's where we need to head, but we're not there yet. I'm going to start wrapping up by telling you that Alzheimer's prevention is not a pipe dream. We are actually conducting trials now in people without any symptoms, but who are at elevated risk for symptoms because of genetic risk. The mutation carriers that we're studying in South America, or older cognitively unimpaired folks who carry 1 or 2 copies of that ApoE4 gene, and also studies in people who don't have symptoms but have biomarker evidence of early pathology. So we actually launched something called the Alzheimer's Prevention Initiative about 13, 14 years ago, when people told us this was absolutely impossible, but this was our case statement. Obviously, there's a huge public health need. By then, we knew that you could see changes in the brain years before symptoms were manifest. We had plausible experimental therapies. We know these biomarkers can track the disease. A detail here that's critically important is, at the time, we didn't have ways of measuring the clinical manifestations in an apparently clinically unaffected person. We needed to figure out that solution in order to get the FDA to create an approval path for preventive therapies. So we did that, which opened up the field for Alzheimer's prevention research. And so this is how we went at it. Let's take people who are cognitively unimpaired, but who are at high imminent risk of symptoms. So in 2012, we launched a major trial in South America that's commanded lots of public attention. So we're immunizing people who carry these mutations but don't have symptoms yet, as young as age 30 as we speak. And it's an absolutely heroic story, and it was labeled by the New York Times and Scientific American as one of the most important science stories in the world. We'll get the readout next year, so fingers crossed on this moonshot. Our next study, you might imagine by now, if I've been making sense, older folks, cognitively unimpaired, but at high risk for symptoms because they carry 2 copies of that ApoE4 gene. Then we did a study, launched a study in people who carry 1 copy of that ApoE4 allele. We're launching a trial this year in cognitively unimpaired people who have elevated brain amyloid. And I'm just going to give you an example of what gets me excited. As we look ahead to the most promising emerging preventive therapies, there's some very cutting edge stuff out there, like going after this apolipoprotein E itself as a target. The studies to see whether those therapies are going to work are going to be very different than the kinds of studies we've been doing before. So we actually successfully recruited a major figure in the pharmaceutical industry to join us, just about 5 weeks ago, Bob Alexander, who's going to lead our efforts in these very early studies. How do you get people who don't have symptoms interested in prevention research? Well, one thing we've done is created an online community that I would invite you to join. The website's at the top, www.endallsnow.org. It's supported by the federal and philanthropic funds, not industry funds. It's an online community of people who are interested in learning about Alzheimer's prevention research. People get content every couple of weeks in a newsletter from world experts. And if people want to learn about prevention research, they can. This has supported roughly 80 studies so far. It's a game changer. And under that umbrella is another game changer where we created a mechanism to allow volunteers to undergo genetic testing for Alzheimer's risk with this ApoE4 test so that they can be matched to studies. It's a complicated issue here. We don't give people the results. That's the practice of medicine. They would only get the results if they were matched with a study that required the presence of an ApoE4 allele and required people to know about that, so that disclosure would occur in the context of a specific study. I actually think that this is an extraordinary story. Just imagine thousands of people looking in the mirror with a cheek swab kit that we mailed them and saying, will I do this so that I can give my genetic information to these scientists who then might match me to a study that might or might not do me any good. I think it's incredible that so many people signed up so quickly, and it speaks movingly to the altruism that a lot of folks have. Getting ready to wrap up. What can we do in the meantime? There are health, exercise and nutritional experts around the world who are doing very compelling research showing that it's very likely that how we treat our bodies, particularly in midlife, will have a big impact on our future brain health. The experts suggest that we may have the ability to reduce risk of symptoms during life by as much as 40% by adopting a heart-healthy lifestyle. So I've listed here the things that have been suggested to matter. And if I were to just say it very simply, get some aerobic exercise and adopt a Mediterranean diet. I've highlighted the aerobic exercise here because that is where the evidence is strongest. We probably should have Medicare paying us to be trained on regular aerobic exercise to stave off possible symptoms of diseases like Alzheimer's disease. And yes, modest consumption of red wine may be protective, but remember, a glass isn't a whole bottle. Just to give you one example, a paper published a couple years ago, an example of maybe 500 or so studies, aerobic exercise had a pronounced effect on somebody's ability to problem solve as they aged. It's probably the best thing you could do for yourself. Okay, so for those of you who don't care about baseball, it may come as a surprise to you that in spring training, despite the pandemic, about half the teams are here in Arizona, the Phoenix area. I'm wrapping up with my last slide, which is a scorecard of where we're at with therapeutics research. I'm not going to say there's been a grand slam, but a grand slam would be one of these prevention therapies working. A home run. I haven't talked about this drug called pimavanserin, but let me mention it briefly since it's on here. Roughly 30 to 40% of persons with dementia of any type will develop frightening hallucinations and or delusions at some point in the course of illness. Very tough to treat, very scary for the patient and the family. The existing therapies have at best modest efficacy and scary toxicity. This medication called pimavanserin has finished a development program showing remarkable effectiveness for relief of psychosis. This drug was on track for rapid approval by the FDA in just a few weeks. And just a couple days ago, the FDA said there's a problem with the file. So nobody knows what that is. So I'm very sad to say that it looks like that approval will be delayed for unknown reasons. Was aducanumab a triple, a home run, a foul ball? You know, that's the one that I mentioned, the convoluted course that it has taken. If I were a betting person, I would bet that the FDA will grant conditional approval of aducanumab, but probably to be used only in a tightly controlled fashion by specialists doing very careful monitoring. Details of some of these others, but oligomanate, so I'm down to the doubles. Oligomanate is a kelp-derived or algae-derived oral agent that alters gut microbiome function. And remarkably, it showed an effect in a large Alzheimer's trial in China, and it is approved for use in China. And a global trial program just launched, including in the United States. So stay tuned. That's the so-called Green Valley pharmaceutical drug. The others listed here are some other monoclonal antibodies that would be follow-on compounds to aducanumab. A sleep aid was found to improve sleep impairment in persons with Alzheimer's dementia. I hinted that this apolipoprotein E gene is emerging as a target. This foul ball, maybe I'm being a bit unfair. A large study called Sprint Mind addressed the question of whether more aggressive versus less aggressive management of hypertension in people who needed to be treated for high blood pressure, more aggressive versus less aggressive treatment conferred protection against emergence of cognitive impairment. The answer was sort of half glasses half full. It was encouraging results, but the primary objective was not met in the study. Knuckleball, which in my book, as an amateur, is that pitch that you can't hit. These blood-based biomarkers are going to be a really, really big deal. Strikeouts, I didn't even get into it, but it's a major, major story. All the oral medications designed to block amyloid misprocessing by blocking those enzymatic scissors, they've all failed. It's a huge blow to the field. Others, there still may be studies done. It would be great if that could be resuscitated, but it remains to be seen. And then on deck is a variety of interesting ideas, some too soon to say. And with that, Emily, I think I will wrap up there and see if there's some questions people want to ask.
So there was one other question. We have about 5 minutes. So if we have time after you answer this question, I have another question for you. The question is about whether the public can encourage the FDA to move on approvals. So whether, writing letters, emails, calls for your triple up there would make any difference.
I believe it would. There are a variety of ways the public can get involved, and the public is invited to get involved if the most common way is if the FDA convenes a so-called advisory committee, or AdCom in geek speak. There was an advisory committee for aducanumab, and there is a period of public commentary. So you can actually sign up to speak, or you can submit something in writing that will be part of the official record. And you can write to the FDA anytime you want, and I think your voice would be heard. And I'm very upset about this pimavanserin wrinkle. I should disclose that I helped the company design their studies and get FDA to buy into the plan and so forth. So it would have been such a help for people suffering these symptoms. I hope the FDA sorts that out quickly.
So one last question I have is, you talked about the APO2 being protective and the APO4 being not what you want to see in your genetic makeup, but there was also a number 3, and I wondered why. I understand there is no one, so we don't have to talk about him. But
Right.
What happened to number 3?
Yeah, so ApoE3 is risk neutral, doesn't increase or decrease risk.
Okay.
So if I carry 2 copies of ApoE3, then how I eat and exercise will be factors, and my age will be a big factor.
Okay. Terrific.
Do we recommend ApoE testing in the clinic? No, we do not, because knowing whether you carry a copy of ApoE4 doesn't tell you whether you will get Alzheimer's disease, when, or what to do about it. But the what to do about it part could change, has changed already, because the what to do about it for thousands of people was, I could get into a prevention trial. So down the road, there will be a role for ApoE4 testing and disclosure in the clinic, but we're not there yet.
Excellent. Well, as always, you are clear on concepts that are incredibly complicated. And so I so appreciate you giving up an hour of your very busy day. And to educate all of us on this incredibly interesting area. And it's so fantastic to hear that there are potential treatments in the future, because I think lots of us have attended Alzheimer's conferences for decades, and the same information was being provided, which is we have stuff that might help with behaviors for a short period of time, but there's nothing on the horizon. So I think this is incredibly hopeful. So thank you so much for your time and efforts.
Well, as they say, thanks for having me, Rachel. I mean, Emily. There might have been another question. I defer to you on time.
No, the questions were thank yous as well for your efforts. And somebody asked if the PowerPoint's available. Again, this is being recorded, so we will certainly have it on our website. We'll be forwarding it to the Alzheimer Institute at Banner, and so they will do whatever posting they do as well. And if anybody else needs the recording, you can always email either myself or the Banner Alzheimer's Institute, and they would be happy to send you the link. Is that accurate, Dr. Theriot? As far as you know?
Sure. Yes, and consider joining that website. It's actually a very high quality product, www.endalznow.org.
Perfect. Well, thank you. Thank you all for staying on. We lost no attendees. And so thank you again, everybody. Take care, and we will see you soon.
Okay. Bye-bye.
Bye-bye.